Fascinating and very urgent topic in plant health. How natural compounds might help us fight one of the deadliest fungal diseases threatening our global food supply. Let's get started. Rice, wheat, and maize, three crops that feed most of the world, are under constant threat from the blast fungus Magnaporthe oryzae. This disease is so destructive that it wipes out up to 30% of rice production globally. And here's the kicker. The fungicides we currently depend on are losing effectiveness because the fungus keeps evolving resistance. So the big question is, can we find safer, natural, and more sustainable fungicides? That's exactly what this study set out to explore. To understand how to stop this fungus, we need to know how it infects plants. It uses melanin to strengthen a special structure called the aprosorium, which basically punches through the plant surface. And it also relies on trehalose and the glyoxylate cycle for energy. So, if we can block the enzymes behind these processes, we can stop the fungus before it even gets inside the plant. The research team screened 39 natural compounds using advanced in silico tools. The goal was simple: find molecules that bind strongly to four essential fungal enzymes, SDH1, 3-HNR, TPS1, and ICL1, and assess whether any of them could serve as future fungicides. These enzymes play critical roles. SDH1 and 3-HNR build melanin. TPS1 helps produce trehalose for energy. ICL1 aids carbon metabolism. Hitting even one of these enzymes is useful, but targeting multiple would be a game changer. Here's how the whole process worked. They collected the compounds, prepared the fungal proteins, ran docking simulations, carried out molecular dynamics for stability, and finally checked physicochemical and bioactivity properties. Let's look at the first target, SDH1. Camptothecin stood out immediately. In Fig. A, you'll see how beautifully it fits inside the active site. Another compound, cryptocin, also bound strongly, shown in Fig. B. This tells us these natural molecules can disrupt melanin synthesis early on. Next, in the melanin pathway we have 3-HNR. Once again, camptothecin was a rock star. Take a look at Fig. A. Ketoviridin also made key contacts with catalytic residues, as you can see in Fig. B. These are exactly the kinds of interactions you want in a fungicide lead. When we move to TPS1, which is involved in energy metabolism, we find a new champion. GKK 1032A2. Figure A shows its tight binding and multiple hydrogen bonds. Camptothecin also showed meaningful binding, illustrated in Fig. B. For the ICL1 enzyme, GKK 1032A2 again demonstrated excellent affinity. Check Fig. A. Camptothecin formed strong hydrophobic interactions too, visible in Fig. B. So by now, a pattern is emerging. Camptothecin and GKK 1032A2 consistently show up as top performers. So, which compounds rose to the top overall? Three of them: camptothecin, GKK 1032A2, and ketoviridin. These compounds don't just target one enzyme, they hit several. And in the world of fungicides, multi-site action is pure gold. When we test whether these compounds look fungicide-like, most pass the Lipinski criteria, meaning they're small, not too greasy, not too polar, and likely to move across cell membranes. This is a strong green flag for further development. Next, the bioactivity test showed that these compounds have biologically relevant activity. Camptothecin especially stood out, but others like HDFO and rockaglale also looked promising from a bioactivity standpoint. Docking is great, but what matters is stability. The molecular dynamic simulations ran for 100 nanoseconds, and the complexes stayed remarkably stable throughout. In Fig, you'll see RMSD, RMSF, SOSA, and RG all showing solid low fluctuation behavior. This supports the idea that these compounds bind tightly and consistently. And when you look at hydrogen bonding patterns in Fig. A and Fig. B, you'll see that especially for GKK 1032A2, the interactions are strong and persistent, similar to what commercial fungicides achieve. Multi-site natural compounds offer a major advantage over single-target fungicides by slowing resistance. In silico, results match earlier lab evidence, especially for camptothecin, GKK 1032A2, and ketoviridin, all of which show strong antifungal potential. The main caveat is that all data are in silico, so in vitro, in vivo and field tests are still needed. Overall, these three molecules stand out as promising eco-friendly fungicide candidates pending further validation. And that's the story. I'd love to hear your thoughts or questions. Happy to discuss.